In this video, I'll be demonstrating the sol gel preparation of zinc oxide nanoparticles. So photocatalysis is a process in which a catalyst material is used to speed up a reaction in the presence of UV light, and is a vital research area because it affords mineralization of toxic organic and inorganic compounds, as well as enzyme disinfection. Semiconductor oxides are attractive photocatalysts because they have high catalytic activities towards various pollutants. Zinc oxide in particular has a band gap of 3.2 electron volts, which is comparable to common photocatalysts, and can be prepared by a sol gel procedure, which involves mixing a hydroxide solution and a zinc acetate solution at 0 degrees Celsius, and then ultimately adding deionized water into the solution in order to precipitate out the zinc oxide nanoparticles. XRD will then be used to confirm whether the prepared zinc oxide nanoparticles match to the reference zinc oxide, and to estimate the crystallite size of the zinc oxide nanoparticles. TEM will then be used to determine the morphology, which is the size as well as the shape of the prepared zinc oxide nanoparticles. And then UVVs will be used to firstly determine the band gap of the zinc oxide nanoparticles, and then to track the degradation of methyl orange and methyl blue dyes using zinc oxide in the presence of UV light. In this part of the experiment, we are going to prepare a sodium hydroxide solution and a zinc acetate solution in absolute ethanol. And we're going to need sodium hydroxide pearls, zinc acetate, 2 labeled Erlenmeyer flasks, 2 magnetic stirrers, 2 vials for sodium hydroxide and zinc acetate for weighing, a beaker, absolute ethanol, a measuring cylinder, a watch glass, as well as a spatula. So we'll start first by weighing approximately 0.14 grams of sodium hydroxide. And to do that, we'll place our vial in the middle of our analytical balance, and then zero it, allow it to read 0, and then transfer the sodium hydroxide pearls into the vial. So we have 0.1457 grams of our sodium hydroxide, which should be fine. And then we'll take that off the balance. And then we'll give our spatula a wipe. And then we'll put the zinc acetate vial in the middle of the balance, as we did before. And then transfer the zinc acetate powder into the vial, approximately 0.55 grams. And before we transfer the powder, we'll then zero the balance to read 0. So we have 0.5582 grams of the zinc acetate, which is also fine. And then we'll take our Erlenmeyer flasks with the magnetic stirrers inside, and then we'll transfer the measured sodium hydroxide into the first Erlenmeyer flask, and then we'll transfer the zinc acetate into the second one. And then we'll transfer the absolute ethanol into a beaker. So since we're going to need around 50, we'll do that for around 100 ml. And then we'll transfer 25 ml of the absolute ethanol in a measuring cylinder. So that's a bit more. So we'll then transfer the absolute ethanol into the first flask and then measure another 25 ml for the zinc acetate. So we'll then transfer that into the zinc acetate. We'll then cover the zinc acetate Erlenmeyer flask with a washcloth and then transfer the flasks onto a heating mantle. So we'll stir the solutions and then heat them up so that the powders can dissolve. So while we are waiting for our powders to dissolve, we'll then prepare an ice bath. So now that the powders have dissolved completely in the absolute ethanol, we're going to transfer the Erlenmeyer flasks into the ice bath so that we allow our solutions to reach 0 degrees Celsius. So that will take around 20 minutes. In this part of the experiment, we are going to prepare the zinc oxide sol, and to do that, we are going to need a warm water bath, a measuring cylinder, deionized water, a plastic dropper, and 4 labeled centrifuge tubes, as well as a stirrer plate. So we're going to start first by placing the zinc solution on the stirrer, and then stir the solution. And then we're going to take our sodium hydroxide solution and use the plastic dropper to transfer the hydroxide solution into the zinc solution. So after we've added all the sodium hydroxide solution into the zinc acetate solution, we'll take the flask off the stirrer plate and then transfer the flask into a warm water bath and let it stand for a few minutes, after which we'll add deionized water. So after we have let our zinc oxide sol to stand for around 5 minutes in the warm water bath, we'll then put that on a stirrer plate and stir the solution, and then we'll take 60 ml of deionized water and then add it to the zinc oxide sol. We'll then transfer the solution into 4 labeled centrifuge tubes. And we'll centrifuge the solutions for 4 minutes using a centrifuge. So once we have centrifuged our solutions for 4 minutes, we'll then decant the liquid into a beaker so that we're left with our zinc oxide gel in the centrifuge tube. And then we'll do the same for the other tubes. And then we'll transfer the zinc oxide solution into the centrifuge tubes over the zinc oxide gel. So now that we have transferred all the zinc oxide solution into the centrifuge tubes, we'll then re-centrifuge again for 4 minutes, after which we'll decant the liquid into the beaker, and then we'll try our zinc oxide gel in an oven for 3 hours at 120 degrees Celsius.